This protocol provides a cell culture platform where the properties of extracellular matrix such as stiffness, protein composition, and cell morphology can be precisely controlled in a cost-effective and facile manner. The method is cost-effective and does not require special training or facilities. It is also compatible with multiple cellular quantification methods such as immunofluorescence staining and Western blot. This technique provides a mechanistic understanding of Schwann cell biology, with additional insight into biomaterial design for nervous system regeneration, and can be applied to any anchorage-dependent cells.
The most challenging portion of this protocol is microcontact printing. Prior to beginning of the protocol, microcontact printing a substrate using a fluorescently tagged protein to visually verify the final printed pattern.
Begin by vigorously mixing the PDMS-based elastomer and curing agents with a pipette tip until bubbles are homogeneously dispersed within the mixture. Then, remove the bubbles with vacuum desiccation. Place a drop of the desiccated PDMS mixture on a square or circular cover slip, and rotate the cover slip on a spin coater at 2500 RPM for 30 seconds. Incubate the cover slip in either an oven at 60 degrees Celsius for 1 to 2 hours, or at room temperature overnight. To prepare a micropatterned substrate, place a patterned silicon wafer inside a circular 150 mm diameter by 15 mm height petri dish, and pour degassed PDMS onto the wafer. Solidify the PDMS in an oven at 60 degrees Celsius overnight. Allow the PDMS to cool to room temperature, then use a surgical scalpel to cut stamps of 30 by 30 mm squares containing the correct patterns, taking care to not damage the silicon.